Hello everyone, and welcome to the fourth and last installment of our recorded lectures for Biology 303 in the fall of 2013. We will continue our discussion of human microbe interactions, and specifically this last installment we will talk a bit more about pathogens and routes of introduction into a host, colonization and growth, and virulence, and then we will talk about a few examples of mechanisms of virulence and cover a few more specific pathogens and their means of virulence. So just to start off, these slides will be posted along with a recorded animation here, so you can look at them separately or view them via the MP4 file. So we'll talk about route of entry into the host, colonization and growth, and then again these mechanisms of virulence. So let's look at the routes of entry into the host. These are very similar to the routes of introduction of the normal flora components into or onto the host cell surface. And we'll talk a little bit about how pathogens mediate this process. And some definitions along the way here are things like virulence. And this is sort of a relative measure of the ability of a given pathogen to cause disease. So what you see over here on the right-hand side of the slide is that you have organisms that are potential pathogens. A host needs to be exposed to those pathogens. We've talked about some mechanisms or routes of exposure, either via direct contact or respiratory inhalation, those sorts of things. Could come from ingestion of either contaminated water or food, those sorts of routes, maybe a puncture directly into the circulatory system. You can imagine a variety of different mechanisms through which a pathogen can come in contact with a suitable or naive host. Once that happens, that pathogen cell has to move into the host cell and then adhere to a suitable infection site. That can be on the skin surface, an epithelial lining, on a tissue surface if the organism is introduced into the circulatory system. And once that adherence occurs, the pathogen needs to sort of invade past that initial adherence site, grow into or past the lining of the tissue or the skin surface, and then be able to grow. Once a growth pattern is established, those certain pathogens might be invasive in that they can move to other sites and cause damage away from the initial site of colonization, or potentially they may be toxic in that they produce bacterial toxins that can be spread by the circulatory system and have either a proximal or distal action that results in pathogenicity in the host organism. So let's look at some of the mechanisms here. The first part is adherence, right? A pathogen has to gain access to host tissues, and to do that has to stick. This is similar to the process we've talked about with viruses. Same kind of thing happens with bacteria. There can be macromolecular interactions that control the specific adherence to different tissue types. For bacteria, this is typically facilitated by extracellular macromolecules, like slime layers and capsules that allow either charge-based interactions or potentially a protein-protein interaction to bind to a particular targeted cell surface or tissue surface. This can also be mediated by fimbriae and pili, and we've discussed these things previously, in that these structures can allow attachment or promote attachment to cell surfaces or other environmental surfaces. Same basic process here, just for a pathogen. Once that occurs, a pathogen needs to invade, sort of move past that site of adherence to access additional resources and also potentially evade detection and removal by the host immune system. This can happen via movement through the circulatory or lymphatic environments. I mean, in terms of getting access to a particular suitable infection site. Once that invasion occurs, the pathogen needs to be able to access necessary resources and be able to grow and increase cell number. Right, and so you can see some shots here of adherence of individual cell types to either an epithelial cell lining on the left, or directly to the bacterial cell surface, or via, say, like a capsule or slime layer to a similar type of host cell surface on the right. Here's a table that sort of describes a variety of these mechanisms of adherence, and again, these are the same kinds of structures, or the same exact structures we've talked about already in terms of envelope components, bacterial envelope components that play a role in attachment to surfaces. And again here, so you just see things like capsule or slime layers, lipoteichoic acids, fimbriae, and then in this case, this list is focused around specific pathogens that use these mechanisms to attach to host tissues. So E. coli uses a capsule or slime layer. Streptococcus pyogenes uses lipoteichoic acids to recognize and attach to host cell surfaces, and so on. Once a pathogen is attached to and initiated a process of invasion and growth in a suitable host, different pathogens have sort of different levels of ability to result in disease formation, and this is known as virulence, or it's a relative term of pathogenicity of different pathogen types. There's different ways to measure this. A typical standardized way to measure this is to look at sort of the lethal dose of a given agent that is required to result in death of a particular test animal. Oftentimes these are done with rats and mice. It's difficult to do these kind of experiments, as you might guess, on humans. And this kind of lethal dose calculation could be done for, say, a protein or a chemical or a carcinogen or what have you, but it could also be done for a microbial inoculum. Right? So fewer number of cells means a lower LD50, which means a more toxic or virulent pathogen, and vice versa for a higher dose. Very virulent pathogens, there may not be much difference in the number of cells that are required to cause death in all exposed individuals versus 50 of them, but then you can imagine less virulent organisms, there might be a more dramatic difference between the number of cells that are required to cause a 50% lethality versus 100%. And here's an example of that. So a highly virulent Streptococcus pneumoniae versus just a moderately virulent Salmonella organism. What you see are the number of cells injected per mouse and the number of mice killed, right, on the y-axis. And so you can see the dramatic difference there. So it takes about 100 Streptococcus pneumoniae cells to kill all of the mice that get exposed. So that's the 10 to the 2 bar there. Whereas it would take 1 million to 10 million Salmonella cells to do the same level of lethality, right? And the LD50 for those two organisms is, somewhere around 50 cells for Streptococcus pneumoniae and maybe 5,000 cells for Salmonella typhimurium. So 2 orders of magnitude difference in the relative virulence between those two different pathogens. There's different terms associated with either limiting virulence or describing levels of virulence. One of those is attenuation. We've talked about that in terms of viruses. You can use that same terminology to apply to a bacterial pathogen. There's also terms known as toxicity. This is typically associated with pathogens that produce a compound that results in disease formation, not due to the cells themselves, but due to some cellular product that may be able to move via the circulatory system to distal locations, or maybe even be produced in one location and then, say, ingested by a naive host and result in a disease state or a toxicity state. There's also this term invasiveness that deals with how well a pathogen is able to adhere to and invade and grow in host cell tissues. This kind of growth can cause damage without producing toxins, just due to damage of the host tissue and acquisition of the resources in that tissue. However, many pathogens use a combination of these kinds of virulence traits, and let's look at one example of that here on the next slide. Here's an example of a particular potentially foodborne or waterborne pathogen, Salmonella. And what you see here shown in this cartoon is a suite of different traits, either associated with the cell envelope, like fimbriae or capsule formation, antigens that maybe inhibit the ability of phagocytes to kill the cell, the many surface-associated different types of carbohydrate or protein-based traits that play a role in adherence and then also potential roles in avoiding, say, immune cell attack. You also see the production of toxins, that enterotoxin that's shown here, kind of that 5 little orange spheres linked together representing a protein that's produced by, or maybe a lipopolysaccharide that's produced by this organism that can result in a disease state or a toxicity to surrounding host cell tissues. Many of these traits may be encoded on plasmids and then have the potential to be shared horizontally, right? And so this slide is a summation of classes of different traits that are common amongst different bacterial pathogens, or at least the suites of things that may be present in different bacterial pathogens that can lead to pathogenicity. So let's talk about a few examples of virulence factors, and so here we'll talk about a couple of different things, including 3 different classes of bacterial toxins, as well as some examples of different pathogens that use these different classes of toxins, and what the disease states look like for those individual pathogen examples. So let's look at enzymes, proteins that can act as virulence factors that have a catalytic activity. For example, hyaluronidase, these are proteins that alter or break down host tissue and provide access to nutrients. This particular enzyme that's shown here breaks down hyaluronic acid, increases permeability of animal tissues, and allows more readily penetration and access to resources in those tissues. Other enzymes might play a role in evading host immune cell responses. So for example, coagulase, one of the traits that a number of you use to identify your unknowns in the laboratory. This is an enzyme that creates a fibrin clot around clusters of pathogen cells and allows those cells to evade immune cell surveillance by the host. There's also envelope components. So we mentioned some of these already. We'll just sort of quickly cover these, things like lipotecoic acids. Another class of cell surface or envelope structures known as protein A can actually bind the constant region of an IgG antibody, and then it makes the bacterial cell look like sort of a cluster of host antibodies. So the cell-specific or the protein-specific binding spots of IgGs would sort of be clustered around the outside surface of the pathogen cell, and then making that pathogen cell more difficult to recognize by the host immune response. The next sort of group of things that we'll talk about are protein, and in most cases anyhow, proteins that are produced by pathogens and exuded outside of the pathogen cell, and then results in some negative series or cascades of events that can result in host toxicity. These are known as exotoxins. So these are proteins that are produced by pathogens as they grow and accumulate or are disseminated by host circulatory systems or lymphatic systems. These are proteins, so they typically can be denatured by heat, so they can be destroyed by cooking or high temperature food preservation. They are also, even though they are proteins, they typically do not result in large degrees of inflammatory responses, even though our immune system is pretty good at recognizing foreign proteins. These particular proteins are small enough that they are not typically well recognized by the host immune response, or at least in terms of inducing inflammation. They can result in memory-based, antigenic, or antibody-based immune responses. The production of them does not result in the sort of humoral response that is sort of the basal inflammatory response of one arm of the immune system. So these proteins are recognized by the cellular immune response, or the memory component of the immune response, but not by the inflammatory component. It's 3 different categories: cytolytic toxins, AB toxins, and superantigens, and we'll talk about each one of those in turn and give an example of different pathogens that produce those classes of toxins. First one we'll talk about are cytolytic toxins. These are proteins, oftentimes called hemolysins, right? So these are things that would produce red blood cell lysis on sheep blood agar. Oftentimes they coalesce to form pores in blood cells and cause those red blood cells to lyse. So these are proteins that essentially affect cytoplasmic membrane integrity that result in cell lysis and death, and that those cells could be red blood cells, they could be other tissues, other cells associated with other tissue types. Oftentimes the lysis is induced by creating a pore that allows sort of relatively uncontrolled or unregulated movement of calcium ions through that pore, resulting in osmotic stress within the host cell and eventually cell lysis. So an example here of an alpha toxin, a cytolytic toxin, different individual toxin molecules can coalesce or self-assemble into a pore structure that gets inserted or inserts itself into the cytoplasmic membrane, and those pores then act as an unregulated channel for the movement of charged ions that can result in osmotic stress on the host cell and eventually cell lysis. Let's talk about another class of toxins. These are exotoxins. There's a couple of different versions of these. Many of these are what are known as AB toxins, and that's that they consist of two subunits, an A and a B toxin subunit. Oftentimes those two subunits have two distinct classes of processes that they catalyze. The B subunit binds to host cell receptors and oftentimes results in the transport or translocation of the B subunit across the cell membrane and into the cell cytoplasm, and that results in some negative downstream process and toxicity in those affected host cells. There's some classes there, are examples, of different AB exotoxins that we'll talk about. Diphtheria toxin, tetanus, and botulism toxins. We'll talk about the organisms that produce those and their mechanisms of action here in just a few moments. The first one we'll talk about is diphtheria toxin, and this is produced by a particular pathogen known as Corynebacterium diphtheriae. The site of attachment and infection for this particular pathogen is the nasopharynx, right? So these sort of essentially the back of the throat and nose. When exposed to this particular pathogen, you get colonization that leads to growth and formation of visible membrane that you can actually see in an infected individual, like just by putting a tongue depressor in the mouth, pressing down on the tongue, and looking at the back of the throat and the nasopharynx region. Oftentimes it can coat the tonsils or essentially have a thin membrane formation over the airway. This results in local tissue destruction, oftentimes due to the production of this Corynebacterium-specific AB toxin. And then the production of the toxin can result in an immune response that results in swelling of the lymph nodes at the back of the throat, and you get this submandibular swelling or bull neck that's characteristic of a diphtheria infection. This is a relatively virulent pathogen, just requires an incubation period of 2 to 5 days, and without treatment, oftentimes results in very rapid onset of death in an infected individual. There are antitoxins for many of these toxins that if you get exposed to these pathogens, you may receive an antibiotic therapy. In addition to that, you may receive an antitoxin therapy just due to the fact that depending on when you start to receive treatment, you may be into the incubation period for a couple of days into the incubation period, and toxin may begin to accumulate, result in tissue destruction, and sort of moving you down the road to death, even though you may be receiving antibiotic therapy. That might not be rapid enough on its own, and so oftentimes that's combinatorial. They'll add in the antitoxin therapy at the same time as antibiotic therapy. Here's sort of an image of a child with a diphtheria infection, and that's sort of a shot of what that bull neck or submandibular swelling looks like. And on the right-hand side, you see this just a cluster of two Corynebacterium cells, sort of characteristic cell association of this pathogen. Let's look at another toxin-forming organism. This is a anaerobic rod, gram-positive rod in the Clostridium genus, and the species is botulinum, or Clostridium botulinum. This particular toxin, if it's consumed, right, blocks the ability of acetylcholine receptor signaling between excitatory neurons and smooth muscle cells. And so what happens here is that this toxin, normally as a signal moves down, an action potential moves down a nerve, when it reaches the end, the terminus of the nerve, that causes the release of acetylcholine that diffuses across the neuromuscular junction, binds to receptors, acetylcholine receptors on the muscle tissue, and causes contraction. The botulinum toxin blocks the release of acetylcholine from the nerve, and therefore the muscles don't ever receive a signal to contract, and results in this sort of descending flaccid paralysis, right? So flaccid meaning lack of contraction of those muscles. This is a very toxic compound, right, where it takes about 5/100ths to 5/1000ths of a microgram per kilogram of body weight of an LD50 in that range, right? So I'm about 80 kilograms in body weight, so we're talking 5, that's 0.4 or so micrograms on the high end of this toxin that would be required to kill about 50% of the people with my body size. Another Clostridium species, Clostridium tetani, also produces a toxin that also inhibits neuromuscular signal transmission. In this case, this toxin acts at a different site, and what it does is it blocks inhibition of release of acetylcholine. So when you get a signal down a nerve cell that releases acetylcholine, typically that acetylcholine release continues until a secondary neuron tells it to stop. Right? That's an inhibitory neuron. What a tetanus toxin does is it blocks signals between the inhibitory neuron here and the excitatory neuron here, such that once a signal for excitation or contraction of muscle is released, that signal continues until the muscle either fails or the nerve cell runs out of excitatory signals, right? So it runs out of acetylcholine. And essentially what happens here for tetanus exposure, as that toxin accumulates, affected muscles begin to contract and remain contracted until the muscle fatigues and fails entirely. And what this looks like in an infected individual is oftentimes flex paralysis, right? So it's not a flaccid paralysis, excuse me, it's sort of an extended, intense, continuous contraction of affected muscles. And what you see here on the left, the bottom left, is an image of a French soldier from World War I. It says tetanus infection. The bottom right is a more modern day or near day photograph of somebody with an intense tetanus infection. And the classic examples of this are all the muscles are contracted. You get this intense U-shaped or arched back. The arms and fists are curled, and you can even see in that drawing in the bottom right is sort of this intense grimace as all the muscles of the face are contracting along with the neck muscles, causing sort of a whole body flex. And as the muscles fail and continually receive this flexing signal, this sort of arching back might also be accompanied by sort of bucking of the body as those muscles continually contract, and some of them may fail and cause short periods of release. It's a very intense and painful way to die in response to a bacterial toxin. Last class of toxins we'll talk about are exotoxins. These are highly antigenic and oftentimes result in intense inflammatory responses. One example of this is an exotoxin produced by Streptococcus pyogenes, results in high levels of inflammation and necrotizing fasciitis of infected tissue. This is where the inflammatory response is so intense it cuts off circulation to the infected tissue and eventually results in necrosis or tissue death. There are also enterotoxins. And these are specific classes of exotoxins that target a specific site in the gastrointestinal tract, the small intestine, and they cause massive secretion and loss of fluids from the epithelial lining and the circulatory system into the intestinal lumen, into the small intestine, and those fluids oftentimes then move into the lower digestive tract, resulting in very watery diarrhea and loss of those fluids by the infected patient. Classic example of this is the toxin produced by Vibrio cholera, the causative agent of cholera. So let's look at how that particular toxin works. Here's a series of images. What's happening here is we see sort of normal movement in the top right panel of sodium ions from the lumen of the small intestine into the circulatory system. So this is one of the roles of the intestinal lining in the small intestine, is to take up sodium from the digestive tract and bring it into the circulatory system. If that portion of the circulatory system is exposed to Vibrio cholera, that pathogen can adhere to the lining of the small intestine, begin to grow, and produce an AB dimeric toxin. The B component of that toxin binds to the GM1 protein that's expressed on the epithelial cell lining, and that binding results in the translocation of the A subunit, or the red subunit, into the epithelial cell cytoplasm. Inside the cytoplasm is where the toxic action of the subunit occurs. And specifically, what happens here is that toxin stimulates the production or the activity of adenyl cyclase, which is an enzyme that converts ATP to cyclic AMP. As that cyclic AMP accumulates in the epithelial cells, that is a signal that blocks uptake, or blocks the transport and the regulated transport of sodium into the circulatory system. So what happens is cyclic AMP builds up in these cells, blocks uptake of sodium. Sodium remains out here in the lumen of the small intestine, and that makes the lumen of the small intestine positively charged, and negatively charged anions in the circulatory system diffuse across this epithelial cell lining and into the small intestine, making the overall small intestine overall heavily charged or ionic, and that pulls water from the circulatory system into this now salty lumen of the small intestine, causing massive water loss from the circulatory system, which is then lost via diarrhea or passage out of the lower end of the gastrointestinal tract. Right? So cholera is a water balance problem induced by a blocking of a normally regulated signal cascade that results in the uptake of positively charged cations like sodium from the small intestine or the lumen of the small intestine. And overall, the toxin action causes the loss of these negatively charged anions that also results in the loss of water. The last class of toxins of so not enterotoxins now, we're talking about endotoxins. Endotoxins are portions of the bacterial cell envelope, specifically a component of the lipopolysaccharide outer membrane of gram-negatives, that when released from lysis of those gram-negative cells results in inflammation, localized inflammation. These are generally less toxic than the protein exotoxins that we've been talking about so far. Again, these are lipopolysaccharides that when released cause an inflammatory response. Here's a table just sort of summarizing some of the actions of these different components of exotoxins, some classes that are classical traits that distinguish them from the endotoxins, and so this sort of suite of traits would be good things to learn for the final exam. This list is just an example of some other organisms that produced different bacterial toxins and their modes of action, the diseases that they cause. You don't need to learn this whole list. It's just an example if you're interested in looking at some of these things. What I will ask you to learn, though, are the specific toxins that we've talked about thus far, and their bacterial producers, right? So which bacterial pathogens produce the kinds of toxins we've talked about in more detail. Those are ones you want to know. This is just some additional detail if you want to take a look at it. And again, that sort of continues here. The last couple of things that I'll just sort of very briefly touch on is risk factors for infection. A lot of these have to do with diet, stress, genetic conditions, age of a host, and whether or not someone's already has been exposed to other pathogens. Right, so the classic kinds of things that we would consider potential for making you susceptible to infection by other pathogens. And that is where we will stop. Good luck with your studying. Good luck on the final. I'll see you during the final during finals week.